So when we're focused on DNA, there are a number of functions for proteins, there are a number of functions for carbohydrates, there are a number of functions for lipids. But when it comes down to DNA, it really just has one key function, and that function is store genetic information, hereditary information, genetic information. So it's really pretty straightforward. We don't really need to worry yet about the purines for pyrimidines. We don't need to know which ones are double ring structures or single ring structures yet. So just the real basics for now. Nucleic acids, it's the overarching category. The monomers are the nucleotides. The polymer are the polynucleotides, or we can just call it DNA or RNA works. This is pretty basic, pretty straightforward. There are three main components to a nucleotide. There is a 5-carbon sugar, either ribose or deoxyribose. There is a phosphate group, and then there's nitrogenous base. And here's the possibilities for nitrogenous bases, right? Adenine, guanine, cytosine, thymine. A's pair with T's, C's pair with G's. It's kind of a double ring structure paired with a single ring structure. To keep the molecules uniform. If I had double rings paired with double rings, it would be wider at some places, and single rings paired with single rings would be narrow. It needs to maintain its position from the other strand, so it's always a double ring, single string matching up. A double ring, single ring matching up with one another. So here's just kind of looking at ribose and deoxyribose. You don't need them. You're not going to have to identify which one's which. It's simply just the location of this hydroxyl group. But you don't need to identify that. Just know that you're a little different from one another. The question on the board will actually deal with that position a little bit. When we look at the DNA molecule, the structural component of the DNA molecule is made from the phosphate group and the 5-carbon sugar. So that they make up the structural component of the DNA. The genetic information is held in the nitrogenous bases. So the phosphate group and the sugar phosphate backbone make up the outside of the ladder, gives it structure. The rungs of the ladder are made up of the nitrogenous bases. DNA has direction. We'll deal with this later. What the direction is, it goes 5' to 3'. But for right now, just understand it has direction. And the DNA molecule is parallel, but it's anti-parallel. The DNA molecules go in opposite directions. So they're parallel to one another, but because they're facing directions, we call it anti-parallel. A couple of things just to notice here when we look at this, right? We can see that the 2 strands are going in different directions, 5 to 3, and then going up here, 5 to 3 going down on the right-hand side. You can see the base pairs matching up, the A's and T's matching up, the C's and G's matching up. Some things here. You can definitely see why a G doesn't match up with an A, or why a T doesn't match up with a C, right? You would have the 2 double rings and the 2 single rings, the width would be affected. But why can't we get a G, a guanine, matching up with a thymine? What's preventing it? On the board, to kind of decipher what that is. So why won't a guanine nucleotide pair up with a thymine nucleotide?
Because the oxygen and the NHs want to be.
Why? So we have these little dots here. What type of bonds are being represented here? What's that? Hydrogen bonds, right? We have polar molecules, so therefore we have hydrogen bonds forming here, right? And between a guanine and a cytosine, how many hydrogen bonds do we form? 3. Between a thymine and an adenine, how many form? So the hydrogen bonding, that really also dictates why you can't get a guanine binding to a thymine. One's really looking to form 3 hydrogen bonds, one's only looking to form 2. Pretty straightforward. The complementary, we already know, right? A's match with T's, C's match with G's. And that's really it for nucleotides. For now, we talked a lot about it later. But that's it. Stores hereditary information. Know the basic makeup of a nucleotide: 5-carbon sugar, nitrogenous base, and a phosphate group. Know just a little bit that the structure is complementary and anti-parallel. That's it for now. there's more to talk about later. So